We are lucky to have Romain Troublé, Executive Director of the Tara Expedition Foundation, who will present the foundation itself and come back on their major explorations. And then, in a second part, Colomban de Vargas, Director of the Station Biologique de Roscoff and Coordinator of the Tara Ocean Expedition. He will use his scientist glasses to present their past discoveries and innovative way of research. Let's watch a quick video now to better know about Tara. So let's welcome on stage Romain Troublé, Director of Tara Expedition.
Hi everyone. I'm Romain. I'm a sailor. I sailed America's Cup twice in my life when I was younger, and I also studied biology also when I was younger. Now I'm more than 40 years old, but I'm heading Tara since 15 years now with Etienne Bourgois, the president of the foundation. We study the ocean. I'm a sailor. We are sailors. We're also scientists, as Jean told you. We are all together on this spaceship Earth across the universe at millions of kilometers an hour, and we barely know a few things about this planet. She's blue. When you ask all the astronauts, and I asked Thomas Pesquet recently, how's the planet from the sky? He said it's blue. It's a blue planet, and 70% of the planet is made of ocean. Huge surface of exchange with the atmosphere, a huge playground for sailors, of course. But if you take off all the water of the planet and you want to put it in a sphere, just to give you an idea of the volume. This is the air we breathe. The atmosphere, we put it in a sphere. The sphere is this size. And if you do the same with all the water of the planet, this is the ocean water. Very, very tiny ball of 1,400 kilometers in diameter. The tiny dots here is the fresh water, and the smaller dots is drinkable water. And our life depends on this small ball of ocean. When you breathe, when you get proteins from the ocean, the CO2 is stored in this ocean mainly. So it's really a life support system. So on Tara, we try to take you in a trip, take the planet, take humanity in a trip on science, discover the planet, try to understand its fate, where we go on this. We try to share that with as many people as we can through the media, through the Google, through the YouTube, through the networks. In many places we go in the world, we've been visiting about 65 countries over the last 10 years, which is kind of big trip. Of course, we share that with kids in the schools, mainly French-speaking school today, but we are now developing curriculums for overseas. We also we also teach the southern countries how to go in the ocean, how to set up scientific boats, how to understand, how to use the richness of the ocean for their behavior, for their economy, for their development. And this is a very strong commitment we've got since 2 years now. And also, as Jean told you, we are putting some scientists on ocean at the UN every 3-4 months to teach as well the people, the kids, the southern countries, and the states, and the negotiators. So over the last 15 years, we did many projects, but we did 4 of them, and I will try to explain you what we did and why we did that. We did one 10 years ago on the climate in the Arctic. We did another one 4 years in a row, fishing plankton, invisible animals. You fish, you don't see what you fish, but you fish every day for 4 years. crazy. Crazy, you think you're really crazy guys. Of course, when you fish plankton across the ocean, you fish a lot of plastic. And this is one of the main disasters we're facing today. This is a reversible disaster. If we do something, we will stop that. But this is a very important disaster. And we also spend a lot of time. Right now, the boat is at the middle of this 2 years expedition in the Pacific. She was this morning in Kim Bay in Papua New Guinea, getting ready for a new survey in this part of the island. And we studied the coral reef ecosystem over all the Pacific for 2.5 years. So this is now. Back 10 years ago, we studied the Arctic Ocean. This is the North Pole. This is the Arctic region. And you can see the North Pole here. And this was done by a guy, crazy guy, who wanted to be the first guy at the North Pole 100th century before, called Nansen. He missed his goal, but we did the same track, and the only one to do it 10 years ago, and we drifted across the Arctic Ocean doing science. I will not go into details, but taking measurements everywhere, down in the water, in the atmosphere, on the ice. We did 6 months of dark, in the dark, 6 months. This expedition lasted 1.5 years. 6 months in the dark, non-stop, pushed by the wind. The ice is crossing the ocean. We did 5,000 kilometers, doing nothing. The world of Tara, this world that you see here, is drifting 10 kilometers per day in the Arctic, pushed by the prevailing winds. And if you look at how it works, you know, it's like tectonic plates. We can have the sound here, maybe put up the volume, please. And the plates in the Arctic, they move not the same speed, and sometimes they crash the boat, and this is real time, and this nothing can we do in our planet can stop that. This is the energy in the Arctic is colossal. This world is drifting 10 kilometers per day. So you study that, and you sit there. This is 6 months day, 6 months dark, and 6 months day again. And this was 10 years ago. Big achievements. We change crew members, we change scientists, we do rotation, logistic rotation to get press people on board and to tell the story of the Arctic, and then again for new nights. This was done 10 years ago, and then back from this upper North Pole area, we said, okay, what's going to be the fate of this ecosystem, the ocean ecosystem in a changing climate? What will be the future? What can we understand? This is plankton. This is drifting across the ocean every day, everywhere, billions and billions of organisms. And Colomban will talk to you more about that. So we went to study this ecosystem across the planet for 4 years on this track, and we collected, in every stop, we stopped and we collected fish, plankton ecosystem, the whole ecosystem, in the same way, repeating always the same kitchen on board, the same filtration, very boring. But you cannot imagine the enthusiasm you can see on board over these 4 years, because the people believed that they were doing something that was completely new. Like Colomban said very often, it's like being in a rainforest with a net, catching birds and new things. So this was a crazy project, including the Arctic Ocean, and Colomban will talk to you more about this science and this knowledge. We did study the ecosystem completely, bacteria, viruses, organisms, algae. We call protist. He's a crazy protist man, Colomban. And we try to understand all this behavior. This is the fish food we talk about. Fish food. The fish, they eat that, and they rely on that. So far, we did a lot of data. It's a big ocean data. We get oceanographic data, molecular data, genomics, images, legal data on every sample, because we were many times in countries that owns these organisms. And we have other connectivity with data centers. And this huge data is kind of the biggest and the first time that we describe an ecosystem completely as a whole. The first time ever it is done. And Colomban will talk to you about now. And these scientists we work with across the world, 25 laboratories from 10 countries, they did publish in Science, did the cover of Science 2 years ago. And this was the start of a crazy adventure in science, and this is still ongoing now, even if we came back from this trip 6 years ago. Doing that, you kind of fish all these organisms, but also you fish a lot of plastic bits that is degrading into the ocean, and that is catching all the life of the ocean. Is also sticking to it as like a rock. It's nice to be at the sun for algae, enjoy the sun, make their life, and they are drifting across the ocean like that. This is a simulation for the Mediterranean Sea. You drop on the model of the Med, you drop 10 particles in every of these 4 main rivers. And you wait for a year to see how they're going. This is a trimester. This is 6 months. 10 particles every day for a year. This is about to be 3 quarters. You can see that particles from the Rhone are going up to Alexandria at the end of the year, which is completely crazy how what we do on land, what we do in our cities, in our daily life, the impact we can have on the ocean and its life. And you can see different patterns, different streams across the Mediterranean Sea. Just to show you the impact. So we did a crazy 7-month journey in the Mediterranean Sea. We collected these plastic things over 7 months, 300 locations, and we are now still studying them. Coming back from this expedition, one of the main ecosystem on the planet, besides plankton, in terms of biodiversity of the ocean, that is storing a lot of biodiversity, 30% of the biodiversity is linked to coral reefs. Very small places, but huge diversity, huge ecosystem, huge biomass as well. So we decided to put Tara for 2.5 years on an expedition to study always the same samples, repeated work over the 2.5 years to sample the reef, its surrounding waters, the organism, the animals. And I can show you here in the video, please, a crazy video showing you the super macro activity of coral reef. You're going to see it's beautiful. And you will understand also the issues with coral reef today. We also talk a lot about bleaching reefs. They get white. But first, look at the beauty of these organisms. So we have divers of Tara diving in many, many places. So far, we did 20 states. These are states, small states. And if you look into details, this is 10 centimeters. This is a millimeter. This is coral. This is an animal. It's like a jellyfish, upside down, fixed on a rock, and it's feeding and it's living. He has like us, we have bones inside. Colors are incredible. You can see these guys getting out at night, usually. Daylight, you do not see them. And so they feed. This is the mouth. This is an animal. They feed. And also, the color you see here is an algae they have in the body that are giving them 90% of the sugar they need to live. 10% is what they eat. And when there is too much stress, too much temperature, too high, they get white. The algae is going away. And if it lasts too long, the animal maybe is starving and dies. And then it's completely covered by seagrass in the ocean. This is climate change. This is what we talk about now everywhere, every year. Even in very remote places, this is 4,000 kilometers from many towns in the world, nearby Pitcairn. All white. So this is not a direct impact. This is really a global impact. And today we have very few understanding of what's going on, how this coral reef is behaving, what are the what are the mechanisms, the process inside to resist climate change, to resist pollution. And we were used to main El Nino events of warming of the Pacific every 6-7 years over the last 50 years. And for the last 3 years, every year we can see bleaching events. Sometimes they recover, and the reef is going back to its state, and sometimes it's too hard and they don't recover. So this will not disappear, but the reef will change. How they will change? The ecosystem will change, the species will adapt and change. How they will change, what will be the fate of this in the future, we don't know. And it's very important for our states, because reef is tourism, economy, fisheries, protection against storms, etc. So the journey now is like that. We are here now in Kim Bay, this place. We have still a lot to go, and come back in France in October in a year, October 2018. Kind of pictures of working down below. Another picture of a very kind boy here. And you can see that we are sampling the reef. We are getting plankton over the reef to see which are the interactions of this. And this will be doing that every day or so. So it's a hard life. It's a hard life. They work a lot. They work a lot and a lot every day on board. They complain a lot because they work too much, but they do an amazing job. And policy-wise, this project is enough, both science, outreach, public, easy to understand, kids are on board. That is very kind of a bizarre project that is attracting Ban Ki-moon twice on board Tara in New York in 2012. And in COP21 in Paris, the boat Tara was in Paris, downtown. And he came again on board with us. And this guy, Eric Carsanti, and Agnès B was the main backers, but also Eric Carsanti was one of the main thinkers of this plankton project that Colomban will talk to you about in a while. Education is key, and I will not go long about that because I'm in short of time. But everywhere the boat goes, we get people on board. Everywhere we came in Paris, there were 10,000 kids came on board with the schools. We do pitches, we do talks in the schools. We try to explain them what we talk about, what we do, give them the feel of the ocean. And everybody is doing it. The scientists, the sailors, everyone. This is really a great time. Good energy, good shots of energy. And the next is, of course, the science is going on, and Colomban will talk to you about that. But also in the future, we're going to go again in the Arctic Ocean. In 2020, we are developing now a polar station, pretty big polar station, to put some scientists on the ice and to understand how this Arctic Ocean is affecting our climate, to understand what's live below the ocean, what is the life there. Nobody knows about it. Nobody has a clue about it. And this will be in 2020. And this device should drift across the Arctic Ocean for the next 20 years, as we plan to do it. So it's going to be a long journey, and it will be the only manned station in the Arctic then. Thank you very much, and I welcome Colomban to come on board. So fasten your seatbelt because you're going to hear science now. So stay calm, okay. Breathe normally, and of course I would like to thank the backers of this project, the scientific backers, but also the private backers that is involved in this project since 10 years. Thank you. Colomban, the floor is yours.
For a nice introduction. Okay, so I'm Colomban. I am not a sailor, but I am a Swiss oceanographer. And I was very lucky to meet actually Romain and also this person that Romain mentioned already, Eric Carsanti. Not sure if this is working or not. Yeah, well. So Eric is an amazing, cell biologist from the EMBL. He spent like 30 years of his life trying to understand how the cell divides and how all molecules within the cell take a shape and auto-organize. So it's the science of self-organization using statistics, mechanics, so it's really the shaping of life with the element of life. And when I met Eric, he showed me this movie, which is a zebrafish. So you can see here all the cell of a little fish dividing. So it's a real fish, okay, under a spin microscope. And you can see this baby fish taking shape under your eyes, all the cells. So it's just amazing. It was the beginning of spin microscopy. And so this is really self-organization of an organism. And when Eric showed me this in 2007, I was thinking, okay, I should follow this guy. And I should. And Eric, besides being an amazing, so he did all this in his lab. They developed the best microscope. And Eric is also a great sailor. He's an amazing sailor, but he never looked, in the ocean of the life in the ocean. But he was brave enough to to think, okay, let's try to understand the ocean that way, and that's always what we had in mind. So basically, we are, today we are really entering this era of ecosystems, and we don't see any organism anymore as a single organism, but as an ecosystem, and mainly human body, right? There are millions now of euros and dollars spent to understand how many microbial genes, because we have many more microbial genes in our body than human genes. We have only 25,000 human genes. We have many more bacterial cells than human cells in our body. So today we really see the human as an ecosystem. And also we start to want to map all the cells of human. I don't know if you have heard about this new project called Human Cell Atlas. It's actually funded essentially by Zuckerberg, by the Facebook Foundation. And where we have the technology to sequence single cell of the genome of single cells, or the expressed genes of single cell. And the idea here is to map all the cells, because we don't even know how many different cells we have in a human body. But all the cells take different shape based on the same genome. They take different shape, and they make our cells, right? So we really have this ecosystemic vision of human. But in fact, what we forget often, and this is very interesting, this is a human cell, this is a macrophage. You have this cell in your blood. Now you have thousands of these cells in your body hunting for bacteria. You have here the erythrocyte, it's another type of cell. And this cell, I just eaten, so it's cleaning your body out of bacteria. So it's really your ecosystem. And what is fascinating, sorry, I was too fast, is that, in fact, if you go to the ocean, you can see this kind of cell. That's an amoeba, for instance, hunting for microalgae here, microalgae. And the resemblance is amazing. And in fact, the composition in salt between your blood and the ocean is strictly almost the same. The content of salt is almost the same. The proportion is different, but we are really a walking piece of ocean. And so I think, the origin of life, is in the ocean. All these organisms have invented all the genes we have and all the shape we have, in our cells. And so we really need to understand these systems, which is much more complex than human, of course. So what we decided in 2008 was really to jump into the big bath and to go to this, like Romain said, to this global ocean, which, in every liter of seawater, you have between 10 and 100 billion organisms, including viruses, in every liter. And the ocean generates all this. It's actually not a blue planet, it's a blue-green planet. You have all this green here, which is photosynthetic cells, like in the tree, but single-cell organisms, which produce half of the oxygen that we breathe on the planet. The ocean is also extremely reactive to global change, so it will feed back on climate very fast. So we really need to understand how this feedback, the resilience of the system, and how it will give us an answer to what we impose to planet Earth. So we decided basically as to, in this era of ecosystems biology, so Eric spent 30 years on this, billions of euros. Now there are many, many projects on this. It's also billions of dollars. So we decided to go directly to this one, to the full system, planet Earth, the biggest system. And the advantage of this system over, classical system that we know, ecosystem that we know better, maybe, as kids, because we see these systems, is that in these ecosystems, the organisms are very big, so it's very difficult to sample these systems. Okay, a forest, it's impossible to sample trees and also the dynamic of these ecosystems is very slow. The climax of the life of a forest is bigger than a human life. While in the plankton, you have a bit of everything. You will see you have a bit of all life, but it's changing, it's reorganizing like the fish, juvenile that I showed you, the baby fish. It's all the time reorganizing. So we think we can understand fundamental principle of ecosystemics. So the beauty of plankton, as I said, is that you don't have the 10th orders of magnitude across which life develop. It stops basically at a few centimeters. And so we developed a protocol to sample plankton from viruses to bacteria to protists to animals. And there is no plants because photosynthesis is done by these protists. We put on the boat, so it was crazy because it was in 2008, we had very little time to equip the boat. So that what was fantastic with Stara was the flexibility, the reactivity, which you can never do, in a classical, in academy. So we equipped the lab, and we were very lucky because it was just the beginning of the high throughput sequencing with the Illumina method. So it was just the time where the price of sequencing was dropping terribly. And so we had, different nets and pumps to sample this full plankton. We also had a very nice rosette, which is an instrument to measure all kinds of environmental parameters. So I will tell you about the data we collected. We collected data in omics, so basically DNA and RNA, expressed genes. Just for the people who like data, we collected about 900 billion Illumina DNA reads. It's more than 90 terabases of raw DNA data from several organismal size fractions from viruses to animals, and from about more than 4,000 plankton communities. This is just, again, the number of data, the number of computing time we have used for now, and the number of analyses that we have done. And as you can see, by far, it's twice, 100 times higher than the next project in ocean systems ecology. So it's really the biggest, by far the biggest, ecosystemic project in the ocean. What is very hard in ecology is to collect, homogeneous data, standardized data across sufficient spatio-temporal scale. So this is for the DNA data. And then this is very original. We used a lot of instruments, flow cytometry. This one we developed, and I will tell you a few words about this one, FlowCam, ZooScan, and UVP, to image as well the plankton. So we took a lot of images of those organisms in the same site at a different depth. And so we haven't almost touched the data, it's very, and so we got, for now, we have 6 million images of plankton. A lot of them are already validated by experts. And we have, which represent more than 30 terabytes of data. We also have done a website, which I think, an instrument on the web, an online image platform, to basically share this image project by project with experts, because we need semi-automated machine learning processes to learn the machine to recognize the images. And so there is no, also there is a gene bank for genes. There is international banks for genes. There is nothing equivalent for image in ecology, in science. So it's really something that we need to think about. OK, now let's go into the data itself, so it will be a bit harder. So we published this wonderful special issue in Science, and of course, we have now, I think we have published 150 papers since 2009, so it's hard to speak about it in 10 minutes. But what we can say from this data, I think, here the 3 papers concern the viruses, the bacteria, and the eukaryotes. Eukaryotes are us, cells with a nucleus. And what is amazing, you don't need to look at the detail, but is that we saturate many of this diversity, meaning that the more we sequence, the less we find new things. And we basically sampled the ocean largely enough, the surface of the ocean, and this was just from the surface, to basically not see anything new. So we pushed the diversity very far away. We sequenced 40 million protein-coding genes for bacteria, but we see most of them, which is fantastic. So we see the limit of this diversity. So we can start to think in terms of ecosystemics. There is just one compartment that we didn't saturate, and this is the eukaryotes. Eukaryotic cells, like our cells, we sequence more and more, and there is many more new. So we are now reaching 150 million eukaryotic genes in this plankton, and it's endless. We are far from saturation. In fact, most of these genes are unknown. They don't look anything that we have in the database. And the other one, we can just attach them to known things, but it's very unsure yet. So this is really amazing, and in fact, I think we are. So just to show you an example of what we can see now with the image data, with using these instruments, we just take picture of the marine snow. The marine snow is all these organisms that fall down to the bottom of the ocean, and which is actually the mechanism that maintain also the atmosphere we breathe. This instrument, with this instrument, we teach the instrument, I mean the data, to recognize this kind of shape, which correspond to giant protists. And we realize that these giant protists are in fact have a bigger biomass than all animals in the plankton in the subtropical and tropical regime here. And they look like that. I mean, no one knows these rhizarians, these colodarians. I think there was like something like 90 species described. These guys, you know, are running the the world. If you dive in the middle of the ocean, you will see tons of these little guys floating. They are beautiful creatures in symbiosis with algae, like corals. They are a bit like piece of corals floating around the world. I think we are almost coming to a new theory of life, of biological diversity. For a long time, we thought that life was, you know, for 3 centuries, that life was in animals and plants, and the diversity was decreasing toward microbes. Then, over the last few years, over the last 3 decades, because people started to sequence the small animals, we thought that the diversity was in the viruses and bacteria. And now that we have a real ecosystemic view of the first system, we see that there is a peak of diversity of genes, of organisms, in the middle, like between, you know, a few microns and 15 microns. That's where really life has complexified. And no one has studied this piece of life, you know. So we speak about diversity. Now we know that diversity is there. And we actually have sequence. We went to the Amazon forest, and it's the same. You find this peak of life, which I call life's middle class, lower middle class, in fact. So these organisms look like that. They are my obsession a bit, protist. So at least I want you to remember this name. And so protists are beautiful creatures, single-celled, so they have very complex genomes. They can build beautiful skeletons. Here you have some coccolithophores. This is always one cell, okay? This is one cell building, so the cell is living inside and is building up these little plates and assembling them outside the cell. You can see how these cells are powerful. This is actually one of these cells building the plate inside some vesicle and extruding the plates. This cell is the most calcifying organism on Earth. You can see how the cell covered the Black Sea here at some time of the year. And these cells, so for instance, this cell, you know, is at the same time a plant and an animal. It can eat, it can photosynthesize, it can build these marvelous, you know, nanotechnology pieces and transform the Earth system, as you can see from the geological record. The shock of Dover here is at 100% coccolithophore almost. So they have an impact, a major impact, probably stronger than the one we had actually today. So the protists are a new world that is now coming into the game of global ecology. And now that what we try also to do a lot is to assemble all this complex data. So we have different layers of data. We have ecological data, we have genomes, genes, organisms, and satellite data as well. So we have different, you know, heterogeneous complex data that we try to put together with graph-based method. And so we did this paper. We built up the first global plankton interactome. And what we saw is that biotic interactions are more important than abiotic interactions. And that, you know, there are much more taxon-taxon interaction in this network rather than taxon environmental parameters. And that cooperation, positive relation, are much more important than exclusive relation, which is kind of a nice lesson, I think, of life. And now you can analyze this. It's going to take years, you know, to understand really what functions are driving this network, this big ecological network. What we have found, for instance, is that, in fact, parasites are incredibly abundant, but nice parasites. You know, these parasites try not to kill their host because they need the host. So parasitism seems to be a big driver of the stability of the network. In fact, we know as humans that, you know, we don't have enough parasites probably. Now there is a medicine that thinks that we should get back our parasites to be healthier. We also start to basically use this network and because this network is really the ecology of a complex system, plankton. Now we try also to mix it to other complex systems like biogeochemical cycles, like the carbon pump, which is a very important fundamental mechanism of the biosphere, or climate. processes. So in this paper, we try to extract the most important component of the network, which are not often the most abundant. Often they are the most connected, or the two fundamental process of the carbon flux that we measured with this instrument I show you before, the UVP, underwater video profiler. And we found that specific plankton communities correlate with carbon export, in particular with the radiolarians, dinoflagellates, and Synechococcus, very important, different species. And we can predict, actually, the carbon export with part of these networks at a very high accuracy. So now, where are we? I think we did half the job, I think. I think we had about, because we sampled, 220 sites, 3 depths, and so we have about, I think, 600, ecosystem in this global body of the ocean. But the ocean is super complex. It's super dynamic. You have a lot of connection. You have this big gyre who have like 300 kilometers large, and it takes them 3 years to cross the Atlantic. And so we need, of course, more expedition, and we need to expand the approach we have in Tara on very, very clever temporal and spatial scales. So Romain told you about. So now we have a new project today, which is called 2C, Tara Ocean Systems, Ecology and Evolution, where we try to. This is the data I just showed you. This is what I call now the symbiome, made of bacteria, protists and animals, metabolism, shapes and behavior. And we need we have a new project basically is to expand the approach we have done over a finer, over different spatiotemporal scales that will allow us to understand the system. One very important aspect is Tara is an incredibly good platform to develop new protocols and to do very, very actually hardcore science. But there is also between 10 and 15,000 sailing boats at any time in the ocean, crossing the ocean. And we started to transfer, as part of the best protocol we have done, to these sailors, which we call now the Plankton nodes in this Plankton Planet project. We have basically extracted a little protocol to collect plankton very easily, and and a little kit that allows them to get a little plankton and send us through the they dry the plankton and they send us just a galette of plankton to the lab directly. And we could show, it was just a test phase, a case study, but it was actually it was super easy to find enough sailors. I mean, we had too many people who wanted to participate. We had 25 crews who sailed the planet in 1 year, and we could collect data from all these dots here, this region. Here is the number of samples of different boats collecting plankton. And we got all this data and we sequenced it for barcoding, for DNA barcode. And here you can see in red the number of new plankton genus that we never found in Tara, in this big data set I showed you, okay? Showing that, in fact, we are reaching saturation, but there is a lot to discover. And if we want to reach a very fine granularity, we need this boat to help, because there is no automated way yet of sampling the complexity of the ocean. The last point I wanted to say is that we need to develop imaging, and we have developed a fantastic approach to automatically image, thanks to Eric Carsanti. And to come back to the beginning of my talk, we developed this new method, this method that was used in human biology to do automatic 3D high-resolution imaging, confocal, of all these cells that I showed you. And it's beautiful because you can really scan the cell through its 3 dimensions, see the chloroplast, the nucleus, the shape, the membrane systems. You can then print the cells, okay, which is beautiful because you can show them. This is plankton for blind people. Huh? You can really show them. It's very important. This cell, it's one of the most abundant on the planet, Earth, right? And so it's so cool to have it in your hand. And actually, I will give you. You can pass it if you want. And now we need to, and also you can see all this symbiosis that these cells are made of. All these cells are congregates, are little ecosystems. They are ecosystems within the ecosystems. So we can study here you can see some cyanobacteria, which are the cells that gave the chloroplast, which are living in symbiosis with some of these organisms. So we can start to understand fundamental process of evolution of this system. The dream is to really understand the rules of evolution, functioning, ecology of one biome on Earth, and basically to understand how different modules we have, their stability, their resilience. If we don't understand this, we will never be able to predict, accurately climate or the future of our, us human body, who totally depend on the biosphere. And there is this guy at MIT, Mick Follows, that does some beautiful, or modeling, including physics and chemistry of the world. He's really a genius. And but the problem is telling me there is no data. Basically, we have the models, everything is working. We just need to include the complexity of life. So I think it's maybe another 15, 20 years, and we will be there. Since 50 years, we know that we have to understand this one. This is really, because it's stronger than us. The dynamics of the of the Earth community will hit the dynamics of the Earth system, and the Earth system is much stronger than we are. That's for sure. So we have to learn how to live in symbiosis with Mrs. Earth, which contain art, actually. So I want to thank very much this was really a first very emotional picture of the beginning of the cruise, who is Eric Carsanti, of course, Romain. You have also Chris Bowler, Etienne Bourgois, who used to be the owner of Tara, now it belongs to the foundation, and myself, and well, it was really the little first nucleus. And thank you very much to you for your attention.
All right, thanks very much, guys. It was an incredibly interesting talk. Some amazing information, amazing visuals, amazing data. We're going to open up the floor for some questions from Googlers. There's a couple of mics up the back there. If you have any questions, you'd like to go up and bring them forward. I have a first question.
Are you still on board? Yes.
Yeah.
Okay.
I might open it up with the first question. Just when I see the imagery of all that, the amazing kind of sea life, the plethora of, all these different kind of colors and things, it's incredible. And then I see the imagery of all the coral bleaching, and it's just really, really impactful. What, in your opinion, can me as a person and me as a Googler do to help with your cause?
What you could do?
Yeah.
There is very simple things to do. I think the problem with ecology is that since decades, we tell people that there's no time. It's too late, or the fate is over, or we have... and by telling that, we really lose a lot of people who don't want to see. If there is no time to do something, you better not want to see it, because otherwise you panic. That's really the panic. And I believe that the change we need in the society as a whole is to change behavior. And this will take generations. And I believe, okay, we have a lot of stress on the planet now. We have a lot of, as you can see, a lot of stress on the ecosystems. I believe we have the time to think ahead. As long as we plan, as long as we know what to do, as long as we do science to understand this planet, I believe that we have time to change in the future. Generation. What you buy every day, when you buy something, you have a huge power on the future of economy. The way you buy things, good things, bad things, healthy things, bad for the environment, bad for the pollution. Really, a choice is very important. The second thing you can do is very cheap, it's free actually. If you have kids, you tell the kids, you try to put them some consciousness about all this. Not the details, of course, but this planet is our land, is our home. How can we do things better in the future? This is easy, and this is like also a trigger for questioning, a trigger for innovating, changing the way we did before, change this way for what we do and do this tomorrow. And of course, the third thing you can do is for when you vote. I don't ask you to vote for ecologists. But keeping in mind this, what they have to say on ecology. Of course, don't vote always for ecologists. I'm not really an ecologist fan. But keep in mind what they propose on this aspect. And this is the way we're going to change slowly the trajectory. And I think this is the ocean is very resilient.
The fifth and very important thing, as Google engineer, you can help us. Basically, we have layers of complex heterogeneous data, global. We have genomes, expressed genes, organisms, images. Then you have the satellite images also that you can connect. So there is a lot of, it's big data. So our dream is to have a Tara Google Ocean platform or something in giant.
This is Google Networks.
Yeah. And we really would dream, actually we enable us to do that because we are too few people. And so if you can help us to do that, and also so there is big data, exploring the big data first, and then there is artificial intelligence for teaching them. Yeah. For instance, a satellite to recognize a gene diversity, or some, you can imagine a lot of things. And I think this is a very good use of artificial intelligence for global ecology. Probably maybe the best use of artificial intelligence.
And if you work really hard, you may have the chance to sail on board. Yeah, really hard.
So we need you, that's for sure.
Yes. Another question?
But
maybe over here on your right, Mark, in the back.
Yes.
Yeah, thank you. This is really fascinating, and it seems like you're straddling lots of worlds, sailing and science. I wanted to ask about generally how you're being supported. It sounds like you have some private support from companies and individuals, but also public support. Yeah. The French government especially seems very interested to follow your work. Could you speak a little bit about especially how governments are helping you and what they're doing to help you?
Oh, it's a real ecosystem as well. We have donators like you and I giving $10 a day, sometime per month or whatever. We have big donors. We have corporates, foundations for French corporate, Swedish, American, and Japanese so far corporations who contribute in sponsorship. They don't have any clue what we do. They don't have any rights on what we collect. It's really sponsorship for knowledge. We also work with collectivities, cities like Lorient, Brittany. In France, we are pretty well supported by the states. And over the last 10 years, since we are with Colomban working on this project, Colomban is working at the CNRS. It's a state laboratory. And
like a postier, or I am
like a postier, like a postman.
No, this is the beauty of the CNRS system. It's super stable. So we have a few people totally fascinated by this project in the CNRS, in the French state, who are going not.
So the French government is supporting this project a lot. They put already $20 million in the research with the Grand Emprunt, with all many different aspects. But we always need more, and we always change the way we do science before. So this is innovation. Innovation is risky, and sometimes it is hard to get the funding for risky innovations.
In a sense, I think the beginning of this started because we were a few people with totally stable work. I cannot be fired from my, even if I don't work. and also there were people like me, and then the private company, and Etienne Bourgois and Agnès B, who were supporting. So this mix between hyperstability and private founders make this actually start. It would have been very hard to start this, with classical academic.
But France took the leadership. The French state took the leadership at the COP21 on climate. We did a lot. France invested a lot in diplomacy about that. And what we do now with STAR is to also teach that the climate, we barely understand it. What's going to be the fate of the ocean? How this will be a reaction? What will be the future? And this is the next step of the COP21, is really to better understand this ecosystem.
In 2013, when we came back, we got a French Investissement d'Avenir funding, which allowed us to organize, to produce most of the data and organize it.
And Cédric Villani was here with us today because we are working now with artificial intelligence. We have so much data that we need that to dig into it. And this is the beginning of a new story. And I hope that Google will be with us on that at some stage.
All right, guys. Thanks very much. I'm afraid we're out of time. Yeah. Again, big round of applause for Tara. Thank you very much.